Welcome to Multiplex Devices webinar on innovative GPCR ion channel assays for the FLIPR Tetra System. This is the second webinar in our ongoing series on this topic. My name is Dave Yamani, and I'm the Senior Director of Drug Discovery Marketing at Multiplex Devices, and I'll be your host today. Today we'll hear two presentations on innovative GPCR and ion channel assays on the FLIPR Tetra System. We're pleased to have Dr. Neil Charter from DiscoverRx Corporation and Carol Crittenden from Multiplex Devices join us today. Time permitting, we will conclude today's webinar with a brief question-and-answer session at the end. Please note that we will be presenting a full-screen view, and there will be a momentary pause between speakers to allow for the presentation control to be transferred to the next speaker. Before we begin, let me invite you to submit questions using the Q&A window on the right-hand side of your screen. To access the Q&A window while in full screen mode, locate the icon tray in the lower right-hand corner of your screen and click on the icon with the question mark. In the Q&A window, you can type in your question and press the send button. Please include the presenter's name so we can properly address your question. Any questions that we do not get to will be addressed privately in a follow-up email message. Our first speaker today is Carol Crittenden, a native of Seattle, Washington. Carol has a BS in biology from Oral Roberts University in Tulsa, Oklahoma. Carol has a background in hematopoietic microenvironment research at the Fred Hutchinson Cancer Center in Seattle. Moving on to cell therapeutics in 1992, a startup biotechnology company, Carol became familiar with cell-based screening methods for oncology compounds. In 2001, Carol established herself as a FLIPR high-throughput screening customer. Returned to California, Carol has been an application scientist at Multiplex Devices since 2002, where she has been an integral part of the instrument development team. As part of the marketing group, Carol has played a major role in developing many new reagents and assays for the FLIPR Tetra System. The title of Carol's talk today is Ligand-Gated Ion Channels, Optimizing a TRPV1 Assay with a FLIPR Calcium 5 Kit. Okay, Carol, I'll just pass the ball to you.
Okay, so I'm going to start my presentation now. So good morning and welcome to our webinar. So I'm going to be talking about TRPV1 ligand-gated ion channels this morning and using the FLIPR Tetra System and our Calcium 5 kit. So for today's agenda, we're going to be talking about why we're interested in this channel in the first place and what it is. And I'm going to talk about assay methods. I'm going to talk a little bit about the FLIPR Tetra instrument and about the kit. And then we'll talk about some results and discussion and a little bit of a summary acknowledgement. So why are we interested in TRPV1 in the first place? Well, it's because it is associated with pain sensation or nociception. So there's a great need for patients to have new and less disruptive methods for reducing pain. And when a TRPV1 channel is activated, it causes changes in cellular calcium concentration. And this makes it low-hanging fruit because we just happen to have an instrument in the FLIPR Tetra System that's very good at measuring the differences in calcium concentrations within the cell. So that's kind of why we're going to be talking about this today. So just a quick review about ion channels. They're found throughout the body. They're part of vision, beating hearts. You've got hearing, taste. And there's a large number of genes coded for different channel proteins. And there's a lot of diseases associated with channelopathies. So it's very fertile ground for drug discovery research. So there's two kinds of ion channels. First one is this, a protein complex that's in the cellular membrane, and they're voltage gated. So when you have a voltage change across the cell membrane, it will cause a pore to be activated or inactivated. The second kind, and this is the kind that we're going to be talking about, is a ligand-gated ion channel, and it's another pore, but instead of being activated by extracellular ligand, and that's what causes the pore to open or close. So TRPV1, it's a ligand-gated ion channel. It's got 6 transmembrane pieces, and then the pore is between the fifth and sixth piece. It was first discovered during experiments with Drosophila phototransduction. They noticed that if the flies were TRP deficient, they'd go blind because the calcium entry into the cell was interrupted. So we talked about that 6 transmembrane. It's also present in neurons and on mononuclear blood cells. So what we're going to be using today is a TRPV1 channel stably transducted into the HEC293 cell. And when that happens, it becomes a non-selective cation channel with a high affinity for calcium. And originally, the transient receptor potential, which is what TRP stands for, was described as the capsaicin receptor on primary neurons. And like we said before, it's associated with pain, sensation, and also it's activated by temperature and changes in pH. And one of the interesting side effects of some of the compounds that block this channel is they actually cause hypothermia. So that's something that they have to really look for when they're looking for new drugs. So this is also called the vanilloid compound. So we're going to be talking about capsaicin, polvanil, and racinoferatoxin today. Those are the 3 main ones that we use. And then the antagonist or blocker we use for capsaicin. There's another part of that group that affects this channel called their endocannabinoids, such as anandamide, which has a similar effect on mood as THC. So it also has nociception capabilities and appetite suppression. So the way I just remember the TRPV1 is capsaicin is the compound that makes chili peppers red hot. So that's one of the ways I remember that. So what we did, we used, again, the HEC293 cells expressing the TRPV1 channel. They were provided to us as part of a collaboration by Chantes Corporation. And these cells have been validated in the manual patch clamp reporting system by Chantes, as well as the Molecular Devices Patch Express automated parallel patch clamp system for seeing that it's a ligand-gated ion channel. And the purpose of our study is to talk about this assay on a FLIPR with the FLIPR calcium 5 kit on the FLIPR Tetra. So the FLIPR calcium 5 kit is part of a family of calcium kits from Molecular Devices, and they were first established about 12 years ago for calcium flex screening for G2-coupled receptors. It was the first no-wash calcium dye, and it uses a masking technology to remove background from the exterior of the cell. So its homogeneous format makes it much easier to handle in a high-throughput screening situation, increases Z factors, and it's compatible with both inherent and non-inherent cell types. And so since it's a calcium-sensitive dye, the cell will fluoresce when it comes into contact with calcium ions. So in the traditional GPCR sense, you've got calcium being released. But in this case, we have calcium released within the cell, but also when the channel is activated, you've got calcium ions coming into the inside of the cell, and then they fluoresce when they come in contact with the dye. So just a little bit about the FLIPR Tetra system and why we're using it. It's because this calcium channel opens quickly, the calcium moves quite fast. We need to be able to record our fluorescence changes while pipetting because it happens so quickly. So that's what the FLIPR does. It's got a pipetter on the top part, and then underneath it has LEDs for fluorescence excitation, and then it has a CCD camera to record the change in fluorescence real time during the assay. So this is the FLIPR Tetra protocol, and it integrates very easily with the software. Just that we're using what we call the calcium set of LED and emission pairs, so we're exciting at about 488, and then the emission filter is the 515 to 575. And then we can control in the FLIPR the power of the LEDs and the camera. This particular experiment was done with the ICCD camera and the FLIPR Tetra instrument, and that instrument or camera is capable of detecting both luminescence as well as fluorescence. assay also works with our EMCCD camera, which does fluorescence only. And the other thing about the software is you can control the aspiration and dispense heights and speeds. And I see that my slides didn't transfer the microliters, so it really isn't 12.5 mils, it's 12.5 microliters. So anyway, that's easily controllable, and it's important when you're optimizing your assay, particularly in this case, because the cells are HEC293. So they are sometimes less adherent to the bottom of the well, and you need to optimize that aspiration and dispense so that you get a good response, but you don't blow the cells off the bottom. So the ScreenWorks software is an easy software for protocol setup, but as well as data analysis. And it allows you to get a one-click EC50, IC50 curve, and so you can do your calculations on the fly when you're getting ready to set up a big screen. You can take a look at Z-factors easily. It has automatic data export. And then the other thing you can do is you can copy and paste these components that you're looking at on the screen easily into your Microsoft Office product or your lab notebook, which makes it really nice. So the first thing we did in the assay was optimize this step. So we thawed the cells and established them in culture following the instructions from TANTest, and they were quite robust and easy to work with. Then the next thing we did is we took a look at which dye would be the best for this, and so we wanted to check the FLIPR membrane potential kit dyes as well as the calcium kit dyes just to see how we were doing. And then we compared it to a competitor kit, because we always do these things. And then we wanted to compare our results also with PathHunter methods. So we wanted to take a look at both agonism and antagonism by the compounds as far as signal window Z-factors and then the EC50 and IC50 combinations. So we grew the cells overnight in 384 Blackwell clear bottom plates with polylysine coating. Well, we put 10,000 cells per well in the plate. And on the day of the assay, we just we formulated the dye and then we added it to the culture medium without having to wash or do anything. And then compounds were formulated in buffer with a 5x concentration because that makes it easier to add on the FLIPR Tetra. And we used the maximum minus minimum relative fluorescence units for comparison, and then I made the graphs using Prism GraphPad. So the first thing we did was take a look at which dye gave us the best response, and we used FLIPR Calcium 5 kit, FLIPR Calcium 4, and then our FLIPR membrane potential red and blue kit. And you can see that in this case, the overwhelming response of signal window came from the calcium 5 kit. Also, there is a calcium 4 kit response there as well, which was quite robust compared to the membrane potential kit. So moving forward, we went on to use the calcium kit. So the calcium 5 response, we compared using 3 compounds. Capsaicin, again, that's the one that makes the red hot chili peppers red hot. And then racinoferatoxin, and then also Olvanil. And we compared the calcium 5 kit response with calcium 4 as well as the introducible 4 direct kit. And you can see in each case, even with slightly different compound classes, that the signal window was the largest with the calcium 5 kit. And across all of them, Z-factors were overall highest with calcium 5, but a good response, and similar EC50s across all the kits. So the other thing we wanted to do was compare that to the PathHunter data. So we used some molecular devices, PathExpress 7000A, automated parallel patch clamp system, to do a comparison of capsaicin. And the sample recordings of the TRPV1 current were evoked by a ramp voltage protocol from minus 100 millivolts to 100 millivolts. And the red line, as you can see, 100 nanomolar capsaicin versus the black line, which is the control. So if we're looking at the EC50 or the dose response curves between the calcium flux on calcium 5 and PathExpress, you can see that the EC50s are very close in concentration. So that's very good. The the other thing we did was we took an inhibitor, which was capsaicin, and ran it using our 3 dyes, as well as a concentration of capsaicin versus capsaicin on the left and Ovanil on the right. And again, you're getting fairly consistent EC50 values across all of them. And the nice thing about the larger signal window from calcium-5 is that if you have a compound that's less clear, which in a screening situation you're going to have, with that larger signal window, it's going to give you more leeway in your assay to take a look at what you're looking at. Also, this was done with an EC80 concentration of capsaicin. However, there are a lot of people now that are starting to screen with EC50s or EC30 concentrations, so that larger signal window, again, is a great benefit in those cases because your overall signal is not going to be that high. So, in conclusion, we found that the calcium mobilization reporter dyes, they're a better choice for monitoring TRPV1 activation than membrane potential dyes because they got a bigger signal window in this case. That may not always be the case, but in this case we saw that. And the EC50 values were consistent between the FLIPR calcium kit assay as well as the between that and the Patch Express assay for the ligand-gated ion channel system. And then for all the agonists and antagonists, we saw that larger signal window up to a 2-fold increase over calcium-4 kit or the FLO4 direct dye. And the pharmacological data was the same across all of them. And calcium-5 kit provided an equal or improved reproducibility for all agonists tested in this experiment. And we measured that with sequence score. Oops. Now there went my last slide. I do want to thank Tantest for their giving us the cell lines and their help with the tissue culture recommendations, as well as Andy Keys and Catherine Parrish. So Dave, that's the end of my presentation, so I'm going to go ahead and pass it back. You go ahead and take it back.
Okay, thank you very much, Carol. Our next speaker is Dr. Neil Charter. Neil is the Director of Profiling and Screening Services at DiscoverRx and is responsible for managing the profiling group that provides compound testing services using DiscoverRx's GPCR and NHR product portfolio. Neil holds a PhD in plant physiology from the Imperial College London, UK. After a postdoctoral research fellowship at the University of California, Berkeley, in the area of neurobiology and biochemistry, Neil joined DiscoverRx in 2002 as a scientist in the R&D department. His responsibilities included developing novel assays for GPCR and kinase drug discovery, including the calcium no-wash plus calcium cell-based assay and the ADP, Quest, and Hunter plus kinase assays. In 2007, he initiated the profiling and screening services for DiscoverRx with the goal to provide customer access to the company's growing cell-based assay portfolio on a service basis. After 4 years, he has developed a team to provide profiling services for nearly 200 GPCRs in multiple assay formats, giving clients the ability to test compounds for multiple GPCR pathways, including arrestin, endocytosis and cyclic-AMP, and calcium second messenger signaling. The title of Neil's presentation today is Dual-Mode PathHunter Beta Arrestin GPCR Cell Lines for Optimized Calcium and Arrestin Detection in the Same Well. Okay, I just passed the ball to you, Neil.
Thank you. Okay. And I get to share the screen.
So just hit the Dual-Mode PathHunter tab on the top.
All right, well, welcome to this to the webinar. Today I'm going to be talking about the use of PathHunter Beta Arrestin GQ coupled cell line for dual-mode calcium and arrestin detection in the same well. DiscoverRx offers a very broad portfolio for GPCR products We have over 500 pre-validated GPCR cell lines available that span multiple platform technologies for a total GPCR solution. We have over 300 arrestin cell lines available for both known, orphan, and ortholog GPCRs. We also have over 120 second messenger cell lines for detecting calcium and cyclic AMP signaling. Our new platform is the internalization assay that provides the ability to measure internalization of the GPCR into the endosomes. So these platforms in combination offer the ability to measure GPCR signaling in multiple pathways, not only second messenger, but also beta arrestin and now internalization. Dave, I'm having problems switching to the next slide. Okay, so the classical model for GPCR signaling is one of linear efficacy. So agonist stimulation of the GPCR creates a signal transduction through the receptor to modulate the concentration of second messengers via a G protein-dependent signal pathway. In addition to activation of the second messengers, agonist stimulation also causes desensitization of the receptor, followed by internalization, and this process is mediated by the binding of beta arrestin to the receptor. In addition to modulating internalization, beta arrestin is also now known to have its own signaling pathway, and this is mediated by beta arrestin-dependent kinases. One of the useful applications of this dual pathway signaling for GPCRs is the identification of novel compounds that can offer bias signaling for one pathway over the other. For example, a compound can activate the GPCR pathway but not activate the beta arrestin, or vice versa. Therefore, understanding the actions of beta arrestin may lead to the development of new drugs and new treatments for human diseases. This is especially important where one particular pathway is important for affecting a disease. The PathHunter technology relies on a system called Enzyme Fragment Complementation. In its simplest form, EFC or Enzyme Fragment Complementation involves the splitting of beta-gal enzyme into two inactive fragments, a large EA peptide and a small pro-label peptide. These in solution can form reconstitute beta-gal activity. For the PathHunter assays, we have developed a novel form of the small peptide tag called ProLink. This has very low affinity for EA, so in solution it has a limited ability to complement and form active enzyme. However, it's able to do so if it is forced into close proximity with EA, and so ProLink can form beta-gal when it is brought together by other proteins. So this is the basis of all the protein-protein interaction assays that we have discovered under the PathHunter platform. The way this technology works in beta arrestin assays is the receptor is tagged with a ProLink tag. This has no effect on the normal expression or signaling of the GPCR, but it offers the ability to have a specific signal related to that tagged protein that has no interference from endogenous proteins. As a result, this this assay is also very universal since majority of GPCRs bind beta arrestin. So on activation of the receptor with beta arrestin, EA and ProLink are brought into close proximity to each other to form active beta-gal, and we can read out the assay using a chemiluminescent substrate. This assay is very useful. It provides a very stoichiometric signal generation and is adaptable to multiple cell lines. Here are some examples of beta arrestin assays developed for a number of GPCRs. The top two graphs show two cell lines that signal through cyclic AMP, a GI and a GS receptor. Notice in both cases these are positive gain of signal assays, simplifying the elucidation of an agonist response, for example. In the bottom two graphs are examples of GQ-coupled receptors, either a type A or type B receptor. So this demonstrates that this type of technology is a universal GPCR solution. Performing the assays themselves is very simple. One simply adds compounds to cells in a plate, allow for the induction of the arrestin binding, and measure the arrestin binding by addition of this one simple reagent and detect it on any simple luminescent reader. Some of the advantages of the PathHunter arrestin assay is its stoichiometric signal generation. This makes it very useful for determining compound efficacy, as well as preserving the rank order of compound potency that one observes for second messenger assays. It is ideally suited for both agonist and antagonist mode assays. It has predictable pharmacology due to the fact that potency is independent of expression levels of the receptor, so you have no concerns about differing levels of expression in these cell lines. Its unique specificity allows it to be used in very complex situations. For example, for serum samples or antibody supernatants, these have been used successfully for detecting both chemokine and bioactive lipids. One of the great features of the PathHunter Beta Arrestin assay is its large assay window. Over 50% of the assays have a 9-fold window or better, and this provides great utility in high-throughput screenings as well as follow-up studies. So today I'm going to be talking about the use of native GQ-coupled PathHunter cell lines for measuring both calcium and arrestin signaling in the same cell. So we have developed over 42 PathHunter cell lines that are naturally coupled to GQ and validated them for both calcium and arrestin. These provide opportunities to perform multiplex screening and profiling. So the idea here is you can run primary screens in both modes to identify compounds that are either selective for one pathway or equally efficacious for both pathways. It is also extremely useful in hit confirmation by allowing you to confirm primary screening results using an alternative technology from the one that was performed in the primary screen. And again, you can use the same cell line to follow up both tests. It's ideally suited for biased ligand discovery to find new compounds that were missed using an alternative signal platform such as a G-protein dependent pathway. It's also very useful in lead optimization and MOA studies because it allows for looking at differences between potency and selectivity between different assay algorithms by comparing the activity in both the calcium and the arrestin pathway. Finally, it is very useful for rescuing intractable targets. But some targets are very difficult to perform using traditional second messenger assays. So it is the able to screen with Arrestin allows you to compare to second messenger results to see what you've been missing. The dual mode protocol for calcium and arrestin detection in using the PathHunter arrestin cell lines is very simple. The arrestin detection reagents are entirely compatible with standard calcium buffers and assays that do not have quenches present. You can perform your calcium assay in the same way that you normally do using DiscoverX's calcium no-wash class reagent. After collecting the calcium data, the arrestin assay can be performed by incubating the assay plate for additional periods of time and then detecting the signal using the PathHunter detection reagent. These assays, or the arrestin assay, can be read on compatible FLIPR or any standard chemiluminescence readers. On the next slide is the kits that are available from DiscoverX for performing these assays. The calcium no-wash plus assay has been optimized for all of the cell lines that are provided for dual readouts in calcium and arrestin. And the PathHunter detection kit also is an optimal reagent for detecting arrestin signaling in these cell lines. Several compatible FLIPR instruments exist that can read both the calcium and arrestin in the same well. These include the FLIPR Tetra with the EM-CCD camera. We recommend that you use no excitation and a high gain to maximize the assay signal. The ICCD camera on the FLIPR Tetra is ideally suited for luminescence detection and can be simply run in the standard luminescent mode. FlexStation 3 instruments are also compatible for both calcium and arrestin readouts. So here is an example of the BDK receptor that has been optimized for both calcium and arrestin readouts. These experiments were performed in the same well, and you can see a very robust performance in both the calcium and the arrestin, with equal potencies in these cases for the two readouts. So this demonstrates the utility of running this type of assay for high throughput screens. We also ran a screen with 10,000 compounds against a muscarinic receptor using both arrestin and calcium readouts in the same cell. For the arrestin assay, we obtained a hit rate of 0.9% versus a hit rate of 2.1% for the calcium assay. 55 hits were confirmed out of approximately 100 in the arrestin assay, whereas 68 compounds were confirmed from approximately 200 hits in the calcium assay. What's interesting to note is that a subset of 11 compounds showed activity in both assays, illustrating that there was a significant number of compounds that showed activity in one mode or the other. So it demonstrates what you can miss by just screening in one assay mode or one assay type. There are two examples where the calcium response for the agonist in these receptors is significantly less shifted from the arrestin response. This poses no problem for running the assay in an agonist mode, but when testing in antagonist mode, it is important to be able to test at the EC80 for both readouts. So the approach we've taken in this test here is to perform the calcium antagonist mode at a lower concentration, and after collecting the calcium data, add a second bolus of agonist to bring the EC80 concentration to the correct concentration for the agonist and the arrestin assay. As you can see here, despite the higher concentrations of agonist required for the arrestin assay, we obtain very similar IC50s for these inhibitors. The dual mode assay is ideal for hit confirmation. In this example here, we took the opioid D1 PathHunter arrestin cell line and ran a selection of compounds in both calcium and arrestin. The calcium assay detected activation not only of the specific overexpressed receptor, but also activation of the endogenous receptors, whereas the arrestin readout was very specific to the tag receptor. The ability to look at these signal pathways in the same cell line is very useful to ensure reliable pharmacology. One of the key features of a dual mode assay is the ability to detect biased ligands. Here is a classic example of the angiotensin receptor, where the biased ligand S2 produces a partial but potent response in the arrestin assay. In comparison, in calcium, the ligand produces a very muted response, but it is significantly right shifted. Here is another example of the dual mode assays being used to elucidate mechanisms of action for a particular pharmacology. In this case, this is looking at insulin release. And the team used mutant receptors, M3 receptors, to establish that calcium mediated a transient early phase insulin release, whereas arrestin modulated the sustained release of insulin after that. Finally, one of the most useful uses for the arrestin platform is for screening intractable targets. Now, some targets are very difficult to screen using second messenger assays. And here are two examples, CXCR7 and C5L2, that signal exclusively through arrestin and do not have a second messenger response. So the arrestin assay in this case is providing additional utility over a second messenger approach. So the benefits of dual mode PathHunter cell lines include a large assay menu of 42 naturally GQ-coupled arrestin cell lines. These stable cell lines express the GPCR of interest with known expression levels that minimize artifacts and allow the use of a single cell all the way through your drug discovery. The ability to measure multiple signal systems enables the discovery of novel biased ligands and develop a more complete understanding of a compound's efficacy, as well as providing a faster, more predictive in vitro experiments. All of these cell lines have been pre-validated for both arrestin and calcium, and this gives you the flexibility to screen or profile in either calcium or arrestin modes. The protocol and reagents have been fully optimized for both assays to ensure large assay windows and robust performance in either mode. As a result, these assays are extremely HDS friendly, providing ease of use and exceptional Z prime, and are highly miniaturizable. Here is the list of the 42 naturally GQ-coupled PathHunter arrestin cell lines that have been developed and validated by Discoverex. Overall, Discoverex offers a very broad technology access to our PathHunter system. We have pre-validated cell lines that include clonal cell lines that are fully characterized, are stability tested, and performance optimized. In addition, we have open systems that allow you to bring in the arrestin technology or nuclear translocation or segregation technology into your own lab and to develop your own assays. If you don't want to do that, we have a broad range of assay-ready kits for both GPCR and cyclic AMP assays that provide all the reagents, including the frozen cells, detection reagents, and plates to perform rapid testing for your gene of interest. We also offer discovery partnerships, outsourcing solutions for custom screening and profiling, and in-depth GPCR and kinase characterization, as well as custom assay development, which is a milestone-based custom service that provides you access to all of Discoverex's ESC technologies to develop novel applications, for example, for orthologs or mutant receptors. So overall, Discoverex offers a very broad platform for your drug discovery needs. We have cell lines, tools, and reagents for over 600 targets spanning 7 platforms, in addition to the custom assay development and profile and screening of outsourcing opportunities for both Discoverex and KinamScan. Here is, if you need more information, here are the contact information, either by website, phone, or email. Please don't hesitate to give us a call if you have questions or want to know more about the product. Thank you very much for joining this webinar. I look forward to hearing your questions.
Thank you, Neil. We will now have a brief question and answer session. Jim Castan, who's assisting me, will read the first question.
Okay, so the first question is for Neil. The first question is, for antagonist mode assays, can you expand on the approach suggested if 2 different EC80 concentrations are required for the dual readout?
Right. So the key thing here is you really need to test at appropriate EC80 concentration. So the unique features of the calcium assay with its incidence readout allows you to perform the antagonist mode of the calcium assay at the appropriate concentration for the calcium assay itself. Once that data is obtained, then the plate can be taken offline and a second addition of EC80 concentration that's appropriate for the arrestin assay can then be added. So that allows you to detect both antagonist responses in the same assay well at different stages in the assay schedule. Okay?
Okay. There's another question. How common is it that customers request dual mode readouts in your profiling service?
That's actually very common. A lot of customers are interested in multiple applications for dual pathway signaling, not only for looking at biased ligands, but also getting confirmation of activity through different pathways. What's clear from the field nowadays is customers are very concerned about missing differences in compound potency over different assay pathways, and that helps them understand the biology or the efficacy of their compounds in their drug discovery program.
And then the interest in testing compounds with second messenger arrestin or internalization readouts?
So The additional internalization assay again provides even more information about how the compounds are acting. So there are several examples of compounds that may induce a second messenger response but not actually affect internalization. So these can have very pronounced effects on the pharmacology, especially when looking to control certain disease types.
Okay. You have a couple more questions. So somebody's asking, do we need to co-transfect the kinase for this kind of ESC assay to make sure that GPCR is phosphorylated upon ligand stimulation?
No, that is handled exclusively by the endogenous GRKs within the cell. So we validated all the cell lines using the Arrestin assay for that type of approach.
Okay, thank you very much. That's it for your questions. So there's a few questions for Carol as well. So Carol, when you say the 384-well plates, do you plate the cells in selection antibiotic media?
No, we don't. We actually remove the selection antibiotic media when it goes from the flask to the plate.
Okay. And then do height and speed of compound addition make any difference in the assay?
Yes, actually it can, just because one needs to get a good mix, so that's one of the reasons why it's a benefit to be able to control that with the FLIPR Tetra instrument, because you can optimize for that depending on your cell line, and so you don't blow the cells off the bottom.
Okay. Another one, why does calcium-5 dye work better than the memory potential dyes?
Because there's more of a response to the changes in calcium concentration versus the change in membrane potential for that one.
Okay, and where does the calcium come from to produce the fluorescent effect?
It can come from either through the pore in the channel, in the ligand-gated ion channel in TRPV1, or it can also come from within the cell.
Okay. There are still, I think we're going to leave the questions open. It looks like there may be a few more coming in. But Dave, I think that's it for right now. If any questions that come in after this will be answered directly to the person asking the question by email. So Dave, back over to you.
Yeah, so thanks, Jim. So as Jim said, if you're still submitting a question, we're going to keep the webinar website open for 5 more minutes to allow you time to send it. And any unanswered questions from today's webinar will be answered privately in a follow-up email message from the presenters. But before concluding today's webinar, I'd like to invite you to visit the danaher.webex.com to see our previously recorded webinars on innovative GPCR ion channel assays on the FLIPR Tetra system. And this concludes today's webinar, and on behalf of our panelists, I thank you for attending, and we look forward to seeing you at future events right here at danaher.webex.com. Have a pleasant day.